guys, I'm going to go ahead and get started. So we're starting a new topic today. Keep in mind that tonight we're going to do lecture, but then you guys are setting up your seed projects at the end of lecture. And so we're going to go over to the lab for that. The first thing that we're going to talk about in cell division is a type of cell division process that happens in prokaryotic cells. And this is called binary fission. So binary fission is a process of cell division that takes place in prokaryotic cells. And what happens in the process of binary fission is you have a cell, which we'll call a parent cell, and then that's going to divide to give rise to two what we'll call daughter cells. And then one really important thing that's going to have to happen during this process is you're going to have to replicate the DNA that's in the cell. And remember that for prokaryotic cells, they typically have one type of chromosome, so you only have to replicate that one type of chromosome and then partition that one type of chromosome into two separate cells during this process. Okay. This produces two daughter cells that are genetically identical to the parent cell, and they're also genetically identical to each other. Okay, so binary fission produces two daughter cells that are genetically identical to the parent cell, and also genetically identical to each other. Another thing where this will come in when we talk about reproduction is that binary fission is a form of asexual reproduction. Okay, so binary fission is a form of asexual reproduction. So this is an example of a single parent giving rise to the offspring. So remember, most prokaryotic cells are single-celled organisms, and if they divide by binary fission, that's a form of asexual reproduction in those organisms. Okay, so what we're going to see is that in contrast to binary fission, the cell cycle in eukaryotic cells is going to be more complicated. And one of the reasons that it's more complicated is the fact that in the nucleus you have different types of chromosomes. And so you're going to have to replicate each of those types of chromosomes. And then you need to partition them correctly. So because there's different types, you need to make sure that the two daughter cells that you produce in the end get the correct types of chromosomes, and then each of them are going to be identical to each other. Okay, so the first thing we're going to talk about is some of the functions of a mitotic cell division. So the first cell cycle that we're going to go through with respect to eukaryotes is a cell cycle that includes mitosis. And there are several functions for a mitotic cell division. In A, this is something that you guys learned in lab last week when we did the discussion of the mitosis and meiosis lab. But one thing that mitosis does is it produces the multiple cells of a multicellular organism. So everyone in this classroom started off as a single cell, and that single cell produced the many cells that make up your body through mitotic cell divisions. Also, mitotic cell divisions are important in wound healing. So when you replace damaged cells, that's going to require pre-existing cells undergoing mitosis to produce new cells. In addition to that, in eukaryotic organisms that are capable of regenerating limbs, limb regeneration also involves mitosis. So the new cells that make up the limb that's regenerated are produced through mitosis. And then the very last one is showing you a single-celled protist. And they're single-celled protists that reproduce through a mitotic cell division. So the protist is a single unicellular organism, and it reproduces through a mitotic cell division. And in that case, that would be also a form of asexual reproduction. Okay, so if you have a single-celled protist that reproduces through mitotic cell division, that is also a form of asexual reproduction. Okay, so what's going to happen in a eukaryotic cell cycle that includes mitosis is we're going to start off with a single cell, and then at the end of the cycle, you're going to end with two daughter cells. And in a cell cycle that includes mitosis, the two daughter cells are going to be genetically identical to each other, and then also genetically identical to the parent cell that produced them. Okay, so overall, what's happening in this process of the cell cycle is you have one cell, and that one cell is going to divide to give rise to two cells. Okay, so some of the things that we have to discuss first before discussing different types of cell divisions that take place in the eukaryotic organism. Are the following, and I'm going to go through them a little bit quickly. You can always, so this is definitions that you should have from a pre-lecture. You can always go back and re-listen to the recording to get the exact definition if you miss it this time. But sex chromosomes, a sex chromosome is any chromosome that's involved in determining sex. Okay, so that was relatively easy. So any chromosome that's involved in determining sex is a sex chromosome. And in humans, that's the X and the Y chromosome. Okay, so the X and Y chromosome in humans are the sex chromosomes. An autosome is any chromosome that's not involved in determining sex. So basically, the rest of the chromosomes besides the sex chromosomes are autosomes. So if it's not involved in directly determining sex, then it's called an autosome. So I previously told you that in humans we have 23 chromosomes in a set, and the first 22 are just numbered or named 1 through 22, and the chromosomes 1 through 22, those are your autosomes. And then the 23rd chromosome in this set is going to be an X or a Y chromosome. Okay? So you have 23 chromosomes in the set. The chromosomes that are named 1 through 22 are the autosomes that are not involved in determining sex, and then X and Y are involved in determining sex. I'm going to skip to the last two because it makes sense to discuss it now. In your bodies, for every cell in your body except for your gametes, and your gametes are going to be, if you're a female, your gametes are eggs, if you're males, your gametes are sperm. With the exception of those cells, all the cells of your body have 2 sets of chromosomes in them. Okay, so they have 2 sets of chromosomes. And you inherited one of those sets from your mother, so you inherited 23 from your mother, and then 23 from your father. So that means that, again, with the exception of your gametes, your cells have 46 chromosomes inside them. Okay, and this relates to what we're going to talk about now, because a diploid cell or a diploid organism refers to having 2 sets of chromosomes. Okay, so diploid refers to having 2 sets of chromosomes. So if I say a diploid cell, again, that cell has 2 sets of chromosomes in it. A haploid cell refers to having 1 set of chromosomes. Okay, so a haploid cell has 1 set of chromosomes. So I previously told you that all the cells of your body, with the exception of your gametes, are diploid. Your gametes are haploid. So if you're a sperm, in sperm you have 23 chromosomes, and in eggs you have 23 chromosomes. Okay, the last 2 things we have to discuss are sister chromatids and homologous chromosomes, and we were kind of introduced to these in the labs that you do the last 2 weeks. But let's say before the cell enters the cell cycle, you're going to have, let's say that's a single chromosome. And so one of the things that you're going to have to do during the cell cycle is replicate all the chromosomes. And what happens in eukaryotic organisms is after this chromosome is replicated, the 2 replicates remain attached to each other. And we call the 2 replicates that are attached to each other sister chromatids. So sister chromatids are the 2 copies of a duplicated chromosome. Oops, not sister chromosome, sister chromatids. Sister chromatids are the 2 copies of a duplicated chromosome that are attached to each other. Okay, so they're going to remain attached to each other, and then later on in the cell cycle they get separated from each other. Okay, this is just vocabulary. It's important in order to understand when I describe the cell cycle. So prior to replicating this chromosome, the word chromosome refers to this. Okay, after you replicate it and the 2 replicates are attached to each other, you call this 1 chromosome. Okay, so it's a little bit confusing, but as long as they're attached to each other, it's still 1 chromosome. So the difference being here, 1 chromosome consists of 2 sister chromatids. Before replication, a chromosome consists of a single chromatid. Okay, and then once you separate them during the cell cycle, then they're 2 separate chromosomes. Okay, so once you separate them, they're two separate chromosomes. But as long as they're attached to each other, they're going to be considered one chromosome. So basically what happens is you're going to replicate all 46 chromosomes in the nucleus, and then initially you're still going to have 46 chromosomes, but every chromosome consists of a pair of sister chromatids. So you would have 92 chromatids total. Okay. And homologous chromosomes are going to be more important when we talk about meiosis. But homologous chromosomes are a pair of chromosomes. This is how we're going to define it for this class. Homologous chromosomes are a pair of chromosomes that carry the same set of genes. Okay, so homologous chromosomes are a pair of chromosomes that carry the same set of genes. We can expand that definition to say that homologous chromosomes are the same size and have the same banding pattern in a karyotype. But the most important thing for this class is to remember that a pair of homologous chromosomes are going to carry the same set of genes. And in terms of your cells, so I told you that the cells of your body have two sets of chromosomes. So that means that your cells have two copies of every chromosome. So you have one chromosome number 1 that you got from your mother, and then you have another chromosome number 1 that you got from your father. Your two chromosome number 1s are homologous chromosomes. Those two chromosomes carry the same set of genes. You got one chromosome from your father, one chromosome from your mother. And then your two chromosome number 2s, those are homologous chromosomes. And then so on. The two chromosome number 3s are homologous chromosomes. Okay? And one of the questions that they asked you in the lab last week is how are homologous chromosomes similar, and then how are they different, because they're not identical to each other. So they're similar because they carry the same set of genes, and the way that you would state this once I tell you what it is they're different from each other because they can carry different what are called alleles. Okay, so what we'll learn, well, I'm going to tell you now, is that a single gene in the population can have multiple forms. Okay, so there can be more than one form for one gene within the population. And you call the different forms of one gene alleles. So an allele would be one form, one variant form of a gene. The one you're probably familiar with if you've taken high school biology is the fact that in genetics you can have a gene that has a dominant allele and a recessive allele. So it's the same gene, it's just that one's going to be dominant and one's recessive. So dads may carry the dominant, but moms may carry the recessive. Okay, so they can carry the same set of genes, but they can be carrying different forms of the same genes, and that's going to make them different from each other. Okay? Yep.
Would you like to say from like white roses compared to red, like when you get different colored roses, or some have like a dominant and recessive?
Well, in the case of the red and white roses, I don't know if that color is determined by dominant or recessive. So I don't know. Purple and white in peas is. But we're going to see an example in Japanese 4-o'clock plants, red and white, and then there's also pink. The two alleles are not going to be, there's not a dominant or recessive. What you're going to see in that case is what's called incomplete dominance. So the alleles, neither allele is dominant in that case. Mm-hmm. Not humans. That's in humans, the height is determined by multiple genes. Okay, so that's it in terms of the definitions that we're going to discuss now. The next two slides are just highlighting again sister chromatids and homologous chromosomes. So before the cell divides into two, you're going to have a single chromosome. You're going to replicate it, and then the two chromosomes after replication are going to remain attached to each other for a while. And while they're attached to each other, they're sister chromatids. We're also going to see that they condense up, so they become more compact. Still sister chromatids. And then once you separate the two from each other, then they're two separate chromosomes. Okay, and sister chromatids are identical to each other. Okay, so that was another question that they asked you in the mitosis lab. They asked you if sister chromatids are identical to each other, and the answer was yes. Okay, so in contrast to that, homologous chromosomes are two chromosomes that carry the same set of genes. So this is showing you two chromosomes. where each chromosome has been replicated and consists of a pair of sister chromatids. And each chromosome carries genes A, B, and D. So they carry the same set, the set being A, B, and D. But then you can see that they carry different alleles for one of the genes, which is the B gene. And so they carry a capital B allele for the on the pink chromosome and a lowercase b allele. It's still gene B, it's just different forms of gene B. And that's going to make those two homologous chromosomes different from each other. Okay, so again, same set of genes, but they can carry different alleles for the same genes. Okay, so now we can kind of discuss the cell cycle, which we're going to divide it up into 4 phases. The first one is called G1, which simply stands for first gap. So it's the first gap phase. So the G1 phase is first gap phase. And then the next phase is the S phase, and S stands for synthesis. And specifically what that refers to is DNA synthesis. So the important event of S phase is DNA replication. So S phase is when DNA replication occurs, and that's what the synthesis refers to. Then you have a second gap phase, which we call G2. So G2 stands for second gap. And then M phase, the M stands for mitotic phase. But the mitotic phase, the M phase, includes both mitosis and cytokinesis. Cytokinesis is the process of dividing the cytoplasm. Okay? So the M phase includes both mitosis and cytokinesis. Also, it's before I get into that, G1, S, and G2 are all part of what's called interphase. So when you guys did the mitosis lab, we had to count the number of cells that were in interphase and then the different phases of mitosis. Cells in interphase are either in G1, S, or G2. Those phases are all a part of interphase. It's also an option for the cell to exit the cell cycle and enter a phase called G0. So you can picture G0 as a cell exiting the cell cycle. And G0 is a non-dividing phase. And cells that have entered G0 have made one of 2 decisions. So if either decided just to postpone dividing, which means that later on they may re-enter the cell cycle and divide again, or the cell has made the decision that it's never going to divide again. Okay, so some cells that enter G0 are never going to re-enter the cell cycle to divide. Examples in your body of cells that are in G0 are your neurons. Also, your muscle cells are in G0, and your retinal cells are also in G0. So those cells are never going to divide again and be sitting in G0. Okay, so those are the different phases of the cell cycle. In terms of when you draw it like this, so you draw it as a circle, so you can see G1 here, S, G2, and M phase. The lengths of the arrows correspond to typically how long those phases take place, or sorry, how long it takes for the phase to take place. So you can see that G1, S, and G2, which make up interphase makes up a majority of your cell cycle typically. And then M phase is only a small portion typically of the cell cycle. So if you remember back in the lab when you guys looked at the root tip cells, they asked you what phase you saw most frequently. The phase you should have seen most frequently was interphase. And the reason that you should have seen that the most frequently is because cells spend most of their time in that phase. So it's most likely you're going to find a cell in that phase versus the other ones because they spend more time in it. The different phases of the cell cycle, there are exceptions and variations in terms of their length. So one example is during development, during early embryonic development. There's going to be cell divisions where interphase is really, really short, and the cell cycle is really, really rapid. because the interphase is really short. But typically, interphase is going to make up about 90% of your cell cycle in terms of time. Okay, the next slide, you don't need to remember the numbers, but this is just giving you how long each phase takes place for a fast-dividing mammalian cells that divides in 24 hours. So an example of that would be a skin cell. That would be a rapidly dividing cell that can divide within 24 hours. This just gives you the typical lengths. So you can see again that interphase is a rather short time period within that 24 hours. It only makes up an hour within the 24-hour time period. The other thing that I want you to remember is that interphase is typically the longest. That's all you have to remember from in terms of the lengths of the different phases. Okay, so what we're going to do is watch this. This is an animation describing the cell cycle, and it'll discuss interphase, and then it'll discuss mitosis, and it'll discuss each phase of mitosis, and then following mitosis, it'll discuss cytokinesis. So this is going to discuss the entire cell cycle.
During interphase of the cell cycle, the genetic material of the cell is found in the form of chromatin and located within the nucleus of the cell, which is surrounded by the nuclear envelope. During the S stage of interphase, DNA replication takes place, and thus the cell leaving interphase and entering mitosis already has replicated its genetic information. In addition to the replicated genetic information, interphase cells that are about to divide also replicate their centrosomes. In animal cells, these centrosomes are formed from centriole pairs. As cells leave interphase and begin prophase, the first stage of mitosis, the chromosomes begin to condense. The nuclear envelope begins to vesiculate, and the centrosomes migrate to opposite ends of the cells to define the two poles. During prophase, the mitotic spindle apparatus begins to form by the polymerization of tubulin proteins. This creates microtubules that emanate from each centrosome. Chromosomes completely condense such that sister chromatids formed from the replicated genetic information and joined at their centromeres are clearly visible. During prometaphase, the nuclear envelope completely breaks down and the mitotic spindle is formed. Polar microtubules extend from both poles and overlap with each other. The sister chromatids become attached to the mitotic spindle via kinetochore microtubules that attach to the kinetochores, which are bound to the centromeres. As prometaphase is nearing its end, the sister chromatids are observed to jerk back and forth between the two poles. During metaphase, the sister chromatids become localized in a region called the metaphase plate. Once the chromosomes have aligned at the metaphase plate, metaphase is complete. When metaphase is completed, anaphase begins. The sister chromatids separate, and each chromatid is now linked to only one pole via a kinetochore microtubule. The kinetochore microtubules shorten, and the individual chromatids, now called chromosomes, are drawn to the poles. Toward the end of anaphase, the polar microtubules push against each other and cause the two poles to be moved farther away from each other. Each daughter cell will receive the same complement of chromosomes as was originally found in the mother cell, thus assuring that each daughter cell gets an identical copy of genetic information. During telophase, the chromosomes decondense and most of the microtubules depolymerize. Portions of nuclear membrane surround each individual chromosome, creating vesicles. The vesicles fuse with each other to create a nucleus containing all of the chromosomes at one pole of the cell. In animal cells, cytokinesis or cell division involves the formation of a cleavage furrow. The plasma membrane is observed to constrict and eventually separates the cell into two daughter cells. The daughter cells then enter interphase, where growth and enlargement occurs, leading to either preparation for another mitosis or differentiation and specialization occurs.
Okay, so that describes the different events of each of the phases. So starting with interphase, then it went to the different phases of mitosis. That also describes cytokinesis in animal cells. So what we're going to do now is just basically do the same thing and talk about the things that you need to remember for each phase of the cell cycle. Okay, so just remember that interphase consists of three phases, which are G1, S, and G2. And the other thing that I want you to remember from this particular slide is that DNA replication happens in S phase. Okay, there's some other things I'll add in later on that happen during interphase, but just write them down then. But interphase DNA replication is specifically going to happen during S phase. So this is when you're going to produce the sister chromatids. So this is when you're going to replicate all the chromosomes, and then the chromosomes are going to remain attached to each other after S phase initially. Okay. Okay, so we're going to start the cell cycle. And one thing that I've included in the lecture questions are basically these slides blank so that you can make flashcards with them. So what I would do is, what we're going to do is we're going to label each phase, and then on the back of the card you can write what happens during that phase or the things that you have to remember, and use that as a study guide. So this one's going to be interphase. Okay, we're just going to describe it as, so this includes G1, S, and G2, but we're just going to describe it as one whole phase kind of together. So one of the things that the cell is doing is it's preparing for cell division. Another important thing that happens during interphase is that the DNA replicates. And again, that happens, you do need to know that that happens specifically in S phase. So then after the DNA replicates, the chromosomes begin to condense. And what that means is that they start to become more compact. Okay, so very early on in the course we talked about the structure of eukaryotic chromosomes, and we talked about a chromosome in terms of the DNA and the proteins that it's bound to, and we talked about the different levels of folding going from a naked DNA molecule to what we call a metaphase chromosome, which is when it's going to be the most highly compact. Okay, that compaction begins in interphase. And then in animal cells, the centrosome duplicates during this phase. And remember from cell structure and function that a centrosome was an example of a microtubule organizing center. So you have one centrosome, and then you're going to duplicate it, so then you'll have two. And then in the animation, they describe something called the mitotic spindle, which is made out of microtubules. All the microtubules that grow to form that mitotic spindle are going to grow from these two centrosomes. And what we're going to see is that after they're duplicated, what they end up doing is they migrate away from each other, and they're each going to define one end of the cell. We'll call them poles, so one pole of the cell. Okay, so those are things that you need to remember for interphase. Then the cell is going to progress into M phase, which we said includes mitosis and cytokinesis. And mitosis is going to be broken up into the following phases, which they went through in the animation, which are prophase, prometaphase, metaphase, anaphase, and then telophase. Okay, so this is going to be prophase. So this is the first phase of mitosis. And in this phase, the chromosomes condense further. And and become visible. And what I mean by that is when you're looking at the chromosomes stained underneath a microscope, they're going to become so compact during this phase that you'll actually be able to visualize individual chromosomes underneath the microscope. So chromosomes condense further and become visible. Okay, another thing that's going to happen during this phase is the nuclear envelope is going to begin to break down. And this is going to finish up in pro-metaphase, but the nuclear envelope begins to break down. And the mitotic spindle is going to begin to form. This is the structure that's going to be made out of microtubules that in an animal cell is going to start growing from the two centrosomes that you made in interphase. And then finally, your nucleolus is going to disappear during this phase. Okay, so these are all the important events that you need to know for prophase. Okay, so then after prophase, the next phase is going to be pro-metaphase. And during pro-metaphase, the nuclear envelope completely disappears. So we started to break it down in prophase, finishes in pro-metaphase. And also the mitotic spindle becomes fully formed. And sort of connected to this, microtubules that are a part of the mitotic spindle, some of them attached to the chromosomes. So microtubules of the mitotic spindle attached to the chromosomes. And this is important because this is going to, the microtubules that attach to the chromosomes during this phase are going to be involved in separating the sister chromatids from each other later on during anaphase. Okay, so then pro-metaphase, the microtubules of the mitotic spindle attached to the chromosomes. Now, it's important to know how the chromosomes are attached to the mitotic spindle, and that's going to be different depending on whether you're talking about mitosis or meiosis. But what happens in mitosis is that this one chromosome is going to be attached to both poles. So remember, each centrosome is going to define a pole, and so microtubules from this pole are going to attach to this single chromosome, and then microtubules from this pole are going to attach to the same chromosome. Okay, so basically each chromosome is going to become attached to both poles during this phase. Okay, so then following pro-metaphase or metaphase, this one's rather easy because what happens in this is the chromosomes align down the center of the cell. So in the human cell, you would have all 46 chromosomes aligned down the center of the cell. And it's also important to distinguish what aligns down the center of the cell when you compare mitosis to meiosis. So for mitosis, again, it's going to be chromosomes that align down the center of the cell. And then we can call the center of the cell the metaphase plate. So you would say that the chromosomes align down the metaphase plate. It's just the same cell between the two poles that are defined by the centrosomes. Okay, so again, in the human cell, during this phase, what you would have is you would have 46 chromosomes aligned down the center of the cell, and each chromosome is attached to both poles. Okay, and we'll talk about how they can attach, or so what they exactly attach to later on. So then after metaphase, this is going to be anaphase. And during this phase, sister chromatids separate from each other. And move to opposite poles. So basically, one sister chromatid is going to move towards one centrosome, while the other sister chromatid moves towards the opposite centrosome. Okay, and this one, once you separate the two chromosomes, once you separate the sister chromatids from each other, then these each become their own chromosome. So if this was a human cell, then you would have 46 chromosomes moving towards this pole, and then 46 chromosomes moving towards the opposite pole. So again, once you separate them, they're two distinct chromosomes. Okay, and then the last phase of the cell cycle the cell cycle. The last phase of mitosis is telophase. And during this, the mitotic spindle is dismantled. A nuclear envelope forms around the chromosomes at each pole. So basically we have two nuclear envelopes forming during this phase. So a nuclear envelope forms around the chromosomes at each pole. The chromosomes begin to decondense, so they previously compacted, now they're going to decondense or become less compact. And also your nucleoli reappear during this phase. So at the end of mitosis, what you're going to have is you're going to have two nuclei, one at each end of the cell. And you haven't divided the cytoplasm yet. Okay? And so that's the next phase. So so mitosis, really, to be really specific, is really just dividing the nucleus. So one nucleus divides into two during mitosis, and then the dividing of the cytoplasm is called cytokinesis. So then so the telophase ends mitosis. Oh, sorry, I forgot the flipboard. So this is anaphase. This should have been telophase. So this here is telophase. Sorry, I forgot to push it forward one slide. And then the cytokinesis overlaps a little bit with telophase. and cytokinesis is specifically the division of the cytoplasm. And how this takes place is going to depend on whether you're talking about an animal cell or a plant cell. And they told you how it happens in an animal cell in the animation, and you guys learned plant cells in the lab, but we'll go through each again. So in animal cells, what happens is on the inside of the plasma membrane, you have a ring of actin filaments. Okay, so just on the inside of the plasma membrane, you have a ring of actin filaments, and what happens is that ring of actin filaments constricts like a drawstring. What happens when this constricts is on the outside you get the formation of a cleavage furrow between what's going to be the two daughter cells. And as the ring constricts, the cleavage furrow deepens, and the cleavage furrow is going to deepen until it pinches the one cell into two cells. Okay, so cytokinesis in animal cells involves a ring of actin filaments and a cleavage furrow, and then that ultimately results in dividing the cytoplasm. Okay, it has to happen differently in plants because plants have a cell wall. And in plants, cytokinesis involves vesicles that are derived from the Golgi apparatus. Okay, so in plant cells, cytokinesis involves vesicles that are derived from the Golgi apparatus. And what happens between the two cells is the vesicles are going to, so you have a bunch of vesicles, and they start to fuse together, and that forms a structure called the cell plate, which you guys learned in lab. Okay, so the cell plate is what's produced by the fusion of all the vesicles. And the vesicles that are fusing to form the cell plate contain the cell wall components. Okay, and then what happens is the cell plate's going to grow outward between the two cells towards the plasma membrane on both sides, and eventually the cell plate will fuse with the plasma membrane on both sides, and that'll end up separating the one cell into two cells. Okay, so do you guys have any questions about how cytokinesis takes place in either animal cells or plant cells? Okay. So two things that we're going to take a closer look at now. One is going to be the mitotic spindle, and the other is the chromosomes when they consist of a pair of sister chromatids. So in terms of the chromosomes when they consist of a pair of sister chromatids, it's important that you know what the centromere is. The centromere, it says this on the slide, is the constricted region where the two sister chromatids are attached to each other. Okay, so the centromere is the constricted region where the two sister chromatids are attached to each other. And then you should know what a kinetochore is. What's going to happen is on the centromere, each sister chromatid is going to have a kinetochore forming on the centromere. So this one chromosome will have two kinetochores. Each sister chromatid has its own kinetochore. And the kinetochore is a protein complex. And the microtubules that attach to the chromosome from the mitotic spindle attach to this protein complex. So the microtubules in the mitotic spindle that attach to the chromosomes attach to the chromosomes through the kinetochore. Okay, so the kinetochore is a protein complex that forms on the centromere, and each sister chromatid will have its own kinetochore, and then the microtubules attach to that kinetochore that has formed on the centromere. Okay, so we also said that one thing that's going to be really important during the cell cycle is going to be the mitotic spindle, which is a structure that's made out of microtubules. We previously said that in an animal cell, the microtubules that make up this mitotic spindle grow from the centrosomes. So remember in interphase, you duplicate the one centrosome, so you have two, and then the two centrosomes are going to migrate away from each other and define separate poles of the cell. So here's one centrosome, here's the second. And then the microtubules are going to grow from both of those centrosomes. And there's actually different types of microtubules within the mitotic spindle. So one type is called astral microtubules. In the picture, it's hard to see it on the screen here, but the astral microtubules are the ones that are in green. And the astral microtubules, it's important that you know the function of each of them. The astral microtubules play a role in correctly positioning the mitotic spindle within the cell. Okay, another type of microtubules are called kinetochore microtubules. And these are the microtubules that attach to the chromosomes. The reason that they're called kinetochore microtubules is because they attach to the chromosomes by attaching to the kinetochore. Okay, so the kinetochore microtubules are the part of the mitotic spindle that actually are going to attach to the chromosomes. And the kinetochore microtubules play a role in separating the sister chromatids. And then the third type of microtubules are called polar microtubules. And when you're looking at this picture, you can see that some of the microtubules coming from this pole overlap with microtubules coming from the other pole. The microtubules that overlap are polar microtubules. Okay, so the polar microtubules are the microtubules that overlap with microtubules coming from the other pole. And these polar microtubules play a role in separating the two poles from each other. So they push the two poles apart during cell division. Okay, do you guys have any questions about the mitotic spindle? Okay. So the next thing that we're going to talk about, let's see if I have the animation still in here. I don't. Okay. The next thing we'll talk about is control of the cell cycle. So a cell isn't simply going through the cell cycle without any type of control. And what that means is that the cell does check to make sure that, one, at the beginning, that conditions are proper for cell division. And then also during the cell cycle, the cell is going to monitor whether things are happening correctly. Okay? And one thing that is involved in the cell cycle control system are these things called checkpoints. And we have to know three of them. So one you can see is called the start checkpoint, and that happens in G1. Then you have one that's called the G2M checkpoint, and then a third one called the metaphase to anaphase transition. Okay, so again, you have the start checkpoint, the G2M checkpoint, and then the metaphase to anaphase transition. Those are three checkpoints that happen during the cell cycle. And essentially what's happening at the checkpoints is the cell is asking certain questions, and if the answer to those questions are yes, then the cell is going to push on to the next part of the cell cycle. But if the answer is no, then the cell is going to halt and wait until that condition is met. Okay, so in the start checkpoint, one of the questions that may be asked is the environment favorable for division? And if the environment is not favorable for division, then the cell is not going to progress into S phase to replicate its DNA. Okay. And then one of the questions that's asked at the G2M checkpoint is, has all the DNA been replicated? Okay, so you can see that the G2M checkpoint happens after S phase, which is where DNA replication occurs. And one thing that you would want to make sure has happened is that all of the DNA has been replicated. If it hasn't, so if the answer is no, then what you're going to do is you're going to pause at the G2M checkpoint and wait for all the DNA to be replicated before you push on to M phase. Okay? And then one of the questions that's asked at the metaphase to anaphase transition is, are all the chromosomes attached to the mitotic spindle? Okay, so that means, is each chromosome attached to both poles of the cell? Okay, and even if you have one chromosome that's not correctly attached, that will pause the cell in that part of the cell cycle. And that just makes sense because if it's not correctly attached, then you're not going to properly separate the sister chromatids from each other. So you do want to make sure that things are properly attached during this checkpoint before you move on to the next phase. Okay, so then the cell is asking certain questions at each of these checkpoints. If the answer is if the answer to the question is yes, then it's going to push on to the next phase. If the answer is no, then the cell is going to pause and wait for whatever's supposed to happen in order for it to happen. Another thing that can pause cells in G1 and also G2. Okay, so cells can pause in G1 and G2 in response to DNA damage. Okay, so DNA damage can cause cells to pause in G1 and G2. And again, that makes sense because if you're replicating the cell, so you're trying to make two daughter cells, you don't want the two daughter cells to receive mutated DNA. So what's going to happen is they're going to pause, they're going to fix the DNA damage, and once the DNA damage is fixed, then they can proceed on to the next phase of the cell cycle. Okay, so the cell cycle control system includes a network of proteins that are involved in monitoring the cell cycle, and that will also be included in pushing the cell cycle forward when it needs to be pushed. And at the center of the cell cycle control system, you have these kinases that are called cyclin-dependent kinases. Okay. So if you remember back to energy enzymes and metabolism, a kinase is an enzyme that adds phosphate groups to molecules. Okay, and they're called cyclin-dependent kinases because if you look at the picture here, the red blob here, that's the cyclin-dependent kinase, and the green blob here is a protein called cyclin. And the cyclin-dependent kinase, if it doesn't have the cyclin bound to it, it's not active. Okay, so in order for it to be active, it has to have the cyclin bound to it. And so that makes it dependent on that cyclin. So the kinase activity of that kinase is not going to be active unless the cyclin is bound to that kinase. And cyclins get their name from the fact that if you look at their levels during the cell cycle, you're going to see that cyclin proteins get synthesized and then they get destroyed. And so they in particular get synthesized during one portion of the cell cycle and get destroyed during another. And then each time the cell enters that phase, that protein is going to be synthesized again. So they show this pattern of synthesis and then destruction. So they show that cycle, so they're called cyclins. And what's going to happen is that, let's see if I have it. The cyclins, when they become active, are going to push the cell to the next phase. So the cyclin-dependent kinase, it adds phosphate groups to its substrates, but that's going to end up resulting in pushing the cell forward through the cell cycle. Okay. Now there's different types of cyclins. You don't need to know them for this class, but I just included them here, and then I will show you. So you have cyclins that are called G1S cyclins, other cyclins that are called S cyclins, and then a third type that are called M cyclins. And then what they're showing you here is when the cyclins are synthesized and destroyed. So again, you don't need to know this detail, but you can see that right here is when the G1S cyclin becomes synthesized and will be present. It'll bind the cyclin-dependent kinase and activate it. And then right around this point in the cell cycle, that cyclin gets degraded. And if it's degraded, then the cyclin-dependent kinase will no longer have the cyclin bound to it and will be inactive. And then right around this point, a different type of cyclin gets synthesized. It binds to a CDK and activates that CDK. Okay, so again, they're called cyclins because they go through a cycle of synthesis and destruction, and they're going to end up activating cyclin-dependent kinases, and then the cyclin-dependent kinases are going to activate certain things that occur during the cell cycle. Okay, don't need to remember anything from this slide. Okay, so you can kind of cross out this slide. This is just showing you the fact that for vertebrates, there's both multiple types of cyclins and also different cyclin-dependent kinases. But in budding yeast, which are unicellular, they have different types of cyclins, but only one type of CDK, which is a cyclin-dependent kinase. Okay, so this is just showing you that when the G1 and G1S CDK become active, they're going to push the cell into S phase. And this is just showing you where each of these cyclin-dependent kinases are going to become active and push the cell through that phase. And what they're showing here is different things that can either activate. So when you look at diagrams of signaling, an arrow means that that's activation. So this means that favorable extracellular environment activates that G1 CDK complex. If you see a symbol like this, where it's a straight line and then one that's perpendicular to it, that means inhibition. And so what that means is DNA damage results in inhibiting these cyclin-dependent cyclin kinases, and that's what would prevent the cell from progressing through the cell cycle. So I said one thing that can halt a cell in the cell cycle is DNA damage, and DNA damage is going to do that by preventing these cyclin-dependent kinase complexes from becoming active. Okay, and you can see that unreplicated DNA or damaged DNA can also then inhibit this cyclin-dependent kinase, and then chromosome unattached to the spindle results in inhibiting that cyclin-dependent kinase cyclin complex. Okay, so that is looking at the control of the eukaryotic cell cycle. Do you guys have any questions about that? Okay, so the last thing, it's not really, we're not going to go into any detail about it, and we've already described it, but apoptosis is a form of programmed cell death, and there is an apoptotic pathway inside the cell, and if this pathway is activated, the cell kills itself. So it's, you can think of apoptosis as a form of cell suicide. And so in terms of when would a cell want to undergo apoptosis, or what could cause a cell to undergo apoptosis, one thing could be unfixable DNA damage. Okay, so if you have a cell in your body that has DNA damage that can't be fixed, you don't want that cell in your body anymore because that could lead to something like cancer or other types of diseases. And so one thing that can induce the cell to kill itself is DNA damage. And that's useful for the organism as a whole. Like the cell kills itself, but then the organism survives because then you're just left with healthy cells. Okay. So one thing that causes apoptosis is DNA damage. Apoptosis also happens as a natural part of development. And so initially inside the womb, you actually have webbed hands. So you have tissue between each of your fingers. But what ends up happening during development is all the cells that make up the tissue between the fingers undergo apoptosis, and then that gives rise to separate fingers. Okay, so apoptosis, one, it can eliminate unwanted cells in the sense that damaged cells, but it is also going to get rid of unwanted cells during development and is a natural part of the developmental process in animals. Okay, do you guys have any questions about apoptosis? Okay, so let's go through some of the lecture questions for this topic, and then we'll take a 10-minute break. Okay, so can someone, the first thing I went through this topic was binary fission. Can someone tell me what binary fission is? So specifically what happens in it, and what type of organisms undergo binary fission? But what happens in it? So it does happen in prokaryotic organisms. One is a form of asexual reproduction. Okay, that's one thing that happens, but overall what happens, you start with, what do you start with, what do you end with? Yeah, right. So you start with one cell divides to give rise to two during binary fission. So number two, can someone, I gave you four functions of a mitotic cell division. Does anyone remember the four? Yep. Right, so animals that are able to regenerate those. Mitotic cell divisions are important for that. What else? Right, during wound healing. What with the face? Right, so in order to produce the multiple cells that make up your body, that requires mitosis. And then what was the last one? And in that case, what is it a form of? So in these organisms, they use the mitotic cell division to do what? Asexual reproduction. So they use it as a form of reproducing.
So the fetus one, could you put development for that, or like what do you have as
Well, you could put, you can't, saying that it's involved in development is not good enough. Saying specifically that it produces the many cells of a multicellular organism is what I'm looking for. So its role in development is producing cells. Because development obviously involves more than just dividing the cells. Then you need the different cells to undergo a specialization process, so you need differentiation. There's also movement that happens, so some of the cells move. Okay, so cells typically spend most of their time in what phase of the cell cycle? Interphase or the mitotic phase? Right, interphase. Remember that typically makes up about 90% of your cell cycle. Can someone describe how cytokinesis is different in an animal cell compared to a plant cell? forms the cell plate? Right, so vesicles fusing from the Golgi apparatus form the cell plate. How does that contrast with cytokinesis in an animal cell? Right. 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 So the cytokinesis in animal cells involves a ring of actin filaments that constrict, forming a cleavage furrow that deepens and will pinch the one cell into two cells. Okay, so can someone quickly go over the three types of microtubules that make up the mitotic spindle, and then also, as you mentioned, each one describe what role it plays in the cell cycle?
Astral microtubules
Right, astral microtubules.
play a role in correctly positioning the mitotic spindle in the cell.
Right,
Kinetochore
kinetochore.
microtubules play a role in separating sister chromatids,
Right,
and polar microtubules separate the tubules from each other.
right. So that's exactly what I was looking for. Okay, this is showing you the two chromosome number 3s from a particular organism. So in this picture, if you were given this picture in an exam, the structures indicated by D are what? Right, A, sister chromatids. 19, this figure is part of a human karyotype. The 3 stands for chromosome 3, so those are the two chromosome number 3s. The best description of what is shown by the entire diagram is what? Right, D, a pair of homologous chromosomes. So the two chromosome number 3s would be homologous chromosomes. They carry the same set of genes. And then I'll show you the picture and then scroll down to the question. So they're asking you what E represents. So in the picture, what does E represent? Right, C, the centromere. That's the constricted region where the two chromosomes are attached to each other. And then what's the name of the complex that forms on the centromere of each sister chromatid? So the metaphase plate is the center part of the cell that the chromosomes align on. It starts with a K. Right, kinetochore. Okay. So 23, the cell cycle consists of several checkpoints at which the cycle can be stopped before restarting. Which of the following is not a function of these checkpoints? Right, supplying energy for the replication of chromosomes into pairs of sister chromatids. So A is true because one thing that can halt a cell at a checkpoint is DNA damage. And so by fixing that DNA damage, that's going to limit the amount of genetic abnormalities that are in the daughter cells. It can also prevent the start of mitosis until all the chromosomes are fully replicated. So we said that happens at the G2M checkpoint. And then also making sure the chromosomes are correctly replicated before nuclear division is complete. And then also ensuring that conditions are favorable for DNA replication and cell division. Okay, 25, chromosomes are replicated during which phase? Right, S phase. And then 26, homologous chromosomes are identical copies of each other. That's false. So how would you describe the difference? Right, they can have different alleles, which are different forms of the same gene. And then I just want to point out again that on this lecture questions, I put flashcards here for you where you can label the phase of the cell cycle, and then on the back of the card, just write down the different things that are different events that you need to remember for each of those phases of the cell cycle. Do you guys have other questions within these lecture questions that you want to go through? Okay, let's take a 10-minute break, and then we're going to start cancer, do the cancer lecture questions, and then start sexual reproduction.